audio, recording first, and then sharing. And I will post this segment of it then, as I said. And we left off right around number 15. Yeah, remember, I actually got to number 15 last time, but because we hadn't really gotten through it and I thought it was incomplete at that point, I told you I would only ask questions pertaining to up to slide 9. So just a little reorientation. Okay, this is a kind of a complex looking thing, and I'm really showing it for a simple reason, basically. It's not in your book, first of all, which is why there's no figure number, but I think it sort of clearly illustrates this one point I want to make. That is that on the inside of the cell, and this is inside on the right side, you have what they have labeled, instead of how I did it, P- for protein ions. They have labeled them A-. But those are the big protein ions that cannot get out. And their presence inside the cell, sort of sets up the whole negativity of the inside negative with respect to the outside. So that's a very key part. Okay, I'll come back to these potassium ions in a minute. But on the outside, I want to show, again, mostly it's sodium and chloride. What's sodium chloride? It's salt. And salt in fluid makes up most of our body fluid, and certainly the interstitial or intercellular fluid in the nervous system between and among the neurons, that's essentially salt water. You'll notice that there are some potassium ions out there. They do leak across. Everything leaks across a little bit, except that the protein ions do not, they're too big. But there's some leakage. And then there's this thing called a sodium potassium pump. And they're showing it here. They're showing that there's this area of the membrane that actively, it's amazing that it works, but it actively pumps sodium out that has leaked across. Now, two things would be driving it to leak across to the degree that it can. Now, so you can't go freely because the membrane is not that permeable to sodium, but it is somewhat permeable. So there will be leakage. Why will there be leakage? In response to the concentration gradient, meaning there's a whole lot of sodium outside in the resting state and very little inside. So it's going to tend to want to leak across. It's going to want to go down its concentration gradient. Also pulling it in would be the electrical gradient. Sodium is a positively charged ion, so it's driven to go inside because it's negative in there. So there are two things kind of pulling it in, and it does leak to some extent. So the membrane makes up for that by actively pumping it out. That's the sodium potassium pump. And it also can pump potassium, that's the K+, that has leaked out. It can pump it back in. So we've got the constant. This nice little diagram shows the concentrations of these 4 ions, inside and outside, rather nicely. And it also reminds us of the presence of a sodium potassium pump that keeps pumping out sodiums that have leaked in. Now, let me ask you a question, and anybody can answer it, or who will. What would happen if you didn't have that sodium potassium pump operating? What would happen to the resting membrane potential? Does anyone have an idea about that? If you just had your cell resting there with its minus 70 millivolts across the membrane, negative on the inside, positive on the outside, and you had the sodium, the Na+, slowly leaking in with nothing to take it out, what would happen to our resting membrane potential?
Wouldn't it just, no, I think I was confused. I was gonna say it would just shoot or initiate an action potential all the time?
Actually,
And-
no, what would happen would it would just zero out. Pretty soon you wouldn't have a resting membrane potential. It'd just be, it would be much closer to zero. So in order
But
to keep it at that minus 70 millivolts, you have to keep that sodium out. Because when it goes in, it's gonna change it. It's gonna make it more positive inside. It's gonna be more positive inside, which means you don't have the resting membrane potential that is needed for an action potential ultimately to be able to occur. Does that make sense? So that we could go into a lot more detail, and if we had a real neurophysiologist, they could do a whole course just on this slide. There's all kinds of stuff and all kinds of things understood and all kinds of medical interventions that go on at this place with medicines, with chemistry. So there's a lot known about it, but we don't need to overkill. I'm just trying to set you up so you can understand the importance of these 4 ions, and also to know what an RMP is, to know what the 2 kinds of graded potentials are, and to know how they interact with each other, those 2 kinds of graded potentials in time and in space, in order to be able to generate an action potential. That's all. And so I don't want us to go deeper. And that's what I found in my 3 questions that I removed. They were going deeper, and it was not worth our effort to try to get there. Okay. All right. So now, hopefully you have a decent understanding of what a resting membrane potential is and how it is maintained through the sodium-potassium pump, and also the differential permeability of that membrane to the different ions. The least permeable to the protein ion. All right, so going to this new slide, let me make it big here. Okay, now we're going to talk about graded potentials. All right, now what is a graded potential? It is a temporary and graded, meaning it could be differentially sized. A temporary graded potential in the electric potential across the membrane. A temporary graded change in the electrical potential across the membrane. And the degree of change is a function of the amount and type of neurotransmitter secreted at a synapse. All right, there are 2 types. One is called an excitatory postsynaptic potential, an EPSP. And that it's excitatory because it tends to move that neuron, the postsynaptic neuron, in the direction of generating an action potential. Remember, we have to have an action potential in order for a neuron to actually communicate with the next neuron in the chain. So if we're generating graded potentials on one neuron, that neuron is not going to generate anything that goes down to communicate with another neuron unless it has an action potential. And in order to generate an action potential, it's got to have a number of these EPSPs being generated on it by different neurons that are synapsing upon it. Okay, when it does its work, when it does its work and with an EPSP occurs, what it does is cause the permeability of the membrane to sodium to increase temporarily. Now I'm going to take us back here. If you take that membrane and you make it be more permeable to sodium, what's going to happen to the sodium? Let me ask you, on the inside or the outside, which one do you want to talk about and what's going to happen to it? Anyone?
So on the outside.
The outside? You want to talk about the outside, okay.
It will be less, it starts to get less positive, so it will become more negative slowly if the sodium is entering the membrane. Is that right?
Well, okay, yes, that's exactly what's going to happen. So if you make the membrane more permeable to sodium, more sodium will enter it. Why will they enter it? Because they're being driven in by their concentration gradient and by the electrical gradient and by the fact that the membrane has allowed them to go in. Now that allowance is only at that little section of the membrane that is postsynaptic. Does that make sense when I say that? So let's say there's one neuron that comes down and forms a synapse on a dendrite of another neuron, and the first neuron generates an EPSP on the second neuron. Where's that EPS going to be? It's going to be right there at that postsynaptic membrane. Now, how does it happen? It happens because the first neuron secreted a transmitter that caused the membrane, the postsynaptic membrane, to open up the channels more for sodium, and then they diffused in. I will look more about at the electricity of that in a moment, but that's the start of our understanding of an EPSP. Does that kind of make sense? Okay. Now, what happens, before we go to IPSP, and I'm going to go back to that other slide, before we go back to ISP, what happens to that EPSP? It just dissipates. The further out away from the postsynaptic membrane, the less the potential change. So if you go out, to the far end of it, nothing happened. It's just at the postsynaptic membrane and then a little bit around it. And the other thing is, if you just watched what happens in time, the EPSP is generated at the moment of the synaptic activity. And if you look, a second later or 2 seconds later, it's restored itself. Sodium potassium pump got everything back to where it's supposed to be. And that EPSP was just a momentary event. And it is less, it dissipates in time, and it dissipates with distance from that point of contact. All right, now let me go back to this slide again, and let's make this one last point here on regarding EPSPs. The potential becomes more positive, or actually less negative. So whereas the resting membrane potential is minus 70 millivolts, it moved to minus 67 or minus 65 or something like that. It didn't go all the way to minus 55, because if it were to get there, there would be an action potential. And any one single EPSP is not capable of generating an action potential. It just can't get the potential change of the membrane that far, from minus 70 to minus 55. It can get a little of the way. But what's got to happen is a whole bunch of EPSPs have to be generated on that postsynaptic cell, let's just call it that neuron, close enough in time and in space that they do get themselves to minus 55. And the place that usually happens, or I would say almost always, maybe it is always, is at the axon hillock. I don't know if I used the term before, did I? I think it's labeled someplace. That's the very, very base of the axon, or where the axon connects up to the cell body and all of its dendrites, right there. That's where it happens. So what's happening is you got all this synaptic activity and all these graded potentials being generated on a cell neuron, a cell body and its dendrites. And if they come close enough in time to summate at the point of the axon hillock, you'll get your action potential. And we'll go more into the action potential, but right now we're just looking at EPSPs and IPSPs. Are you okay with the concept of the EPSP? I'm looking for head shaking. I'm looking for thumbs up. I'm looking for anything other than blank stares. Or do you have any questions about the EPSP before we go on?
I'm having
Georgina, yes.
As far as, so if it's in the inside or the outside, it tends to matter, right? Because doesn't it go from high to low? Like if there's a lot of positives, right, or negatives, it doesn't want to level. As far as it being excitatory, doesn't the inside want to go to the outside if it's, or the opposite, vice versa?
I'm not quite sure what you mean by doesn't the inside want to go to the outside?
Because we're talking about the Na, right?
Oh, the Na, okay, yes.
Yeah. Sodium, right?
Uh-huh.
So you were talking about if it's in the outside or in the inside or the outside, and I think I think it was,
If what's on the inside or the outside, the sodium?
yeah, the sodium.
Okay,
Yeah.
the sodium, yeah.
Yeah. So I'm trying to figure out, what do you mean by that? Does it not matter anymore?
Okay, well, let's go back here and let me take a stab at your question. You see how close I come. So on the resting state, remember that's the state where the potential is going to be minus MV 70 millivolts across the membrane. In the resting state, here's our balance of things. And when the EPSP is generated on this membrane, what it does is it causes that membrane to let sodium in. It becomes more permeable to sodium. So at that point, when that happens, now the inside is less negative than it was. How negative was it? Minus 70 millivolts. How much less? Well, some, I said, how about 68 or 67? What it doesn't do is get it all the way to minus 55. That's less negative too, but it's much more less negative. No, is that true? Yeah, it's much more less negative. Oh, that's a horrible sentence. Than a single EPSP is capable. It can only move it a little bit of millivoltage. And then if you just sit there and wait, what happens? Well, it all restores itself, so it never really gets very far. You got to have a whole bunch of these occurring in a small enough period of time that you get that axon hillock to minus 55, and then you get the action potential. We'll talk about what happens to that membrane when you have an action potential in a moment. Okay, does that work, Georgina, at all? Okay, good, thank you. All right, so let's go now to the inhibitory postsynaptic potential. The inhibitory postsynaptic potential has exactly the opposite effect. It tends to cause greater negative potential across the membrane. Now, how does it do it? Go back to here. Well, let me show it here. It increases the permeability to the K+, which is potassium. Na+ is sodium, K+ is potassium. It becomes more permeable, not to sodium, but to potassium, K+. Now come back here. Where is all the K+? It's on the inside. So if you make this more permeable to it, it's going to want to leak out here. And it's only driven by the concentration gradient, not by the electrical gradient. So it's not as driven, but it is driven by the concentration gradient, and it will leak out. And when it leaks out, it's taking a positive charge from the inside to the outside and causing the inside to become a little more negative because it has that one less positive charge to help it out. All right, so it has exactly the opposite effect. Now you could sort of think ahead. What would happen on a postsynaptic cell if you had two synapses real close to each other on some dendrite on that cell, and simultaneously one generated an EPSP and the other one generated an IPSP? What do you think is going to happen?
They cancel out.
They cancel out. Yeah, nothing's going to happen. You'll get the change right at the postsynaptic membrane. But if you go out from it a little bit, say, well, what happened now? Nothing happened because they canceled each other out. Okay, now how does it work? The way it works is by the chemical nature of the transmitter. The particular transmitter that is located in the boutade of the presynaptic cell, the chemistry of it is such that it can do one and only one thing for its entire life. It can generate either an EPSP or an IPSP. That's it. And so how do you get all these EPSPs and IPSPs adding up and making action potentials? Well, it's because if you look at any one cell body with its dendrites, it has a whole lot, thousands of synapses on it, some of which make IPSPs and some of which make EPSPs. You begin to get, I mean, it's mind-boggling. You can't really figure it out, but it begins to give you the degree to which complex information can be processed, because isn't this all just a whole bunch of switches? That's what a computer is. It's a whole bunch of switches that are on or off. I don't know much about it, but I think that's kind of what it is. And that's what your brain is. It's a whole bunch of switches. How many? Well, we have 80 billion to 100 billion neurons to begin with. Each of those has many thousands of synapses on it. So it's mind-boggling, the numbers we're talking about. And the amount of information and the complexity of information that can be processed by the brain. At a moment like this, you feel like your brain can't process anything, but you know, it is. All right. Any questions on EPSP or IPSP? Yes.
So are there EPSP and IP highways in our system?
I wouldn't describe it. You mean like, are you saying like certain named tracks of a pathway, something that you would say, you know, like your facial nerve or something like that, that are just generating EPSPs and IPSPs?
Yeah, because it seems that they do both,
Yeah.
but then they don't.
Well, okay. Now, when you come down to the level of a motor neuron that goes actually out to the muscle, and that's got a synapse too. It's got a bouton, it's got a synapse, and the synapse is on the muscle. And the muscle is not a nerve, but the membrane acts like a nerve, so it responds in the same way. Those are all EPSPs. But inside your brain, you don't have tracks that just do EPSPs and tracks that just do IPSPs. It's wired up more complexly than that. And we will have obviously tracks of nerve that we're going to give a name to and describe the function. But we're not going to then talk about how some of the neurons are generating EPSPs and some are generating IPSPs, but that would be a fact that some of them are. That's how action potentials get generated.
Well, I was thinking about the words retrograde and antegrade.
Amnesia?
No, antegrade, like there's a system in our nervous system. I think I read it in chapter 2 or 3. And we have some things going retrograde towards the nerve cell and some things going anterograde.
Okay. I don't know what that is. If I knew what the things are, maybe I could make a stab at the question. But if you could find that for me, I can probably respond. But yeah, I'm not sure what it is you're referring to just from that. Sorry.
Right. It was just that I was thinking we do have highways. We're not like plants.
Oh, you cut out. You cut out. I didn't hear that part.
Oh, yeah, I was thinking that we're not, units have a phylum and things go down. And I was thinking maybe in humans, we're like that too. We have highways of efferent and afferent nervous parts.
We do have that.
But maybe we do.
Oh, yes, certainly. Yeah, we do have pathways.
Work like highways.
Yeah, we have pathways that carrying information all in one direction. But then when you get up to the level of where the synapses are occurring, you know, like when we show a 2-neuron chain, that let's take I kind of gave this example last time, but we haven't really gotten into it. You take a motor neuron in the motor strip, and it goes, if it's one that's going to ultimately cause your toe to move, it's called a cortical spinal neuron. It goes all the way from the motor cortex all the way down to the bottom of your spine with no synapses until it gets down there. And then it synapses on a lower motor neuron that actually goes out to the muscle. So it's 2 neurons, a 2-neuron chain from your motor cortex down to your muscle, toe muscle. Okay, and it's carrying information all in one direction. So I think that's responding to what you're talking about. It's got one function. However, that synapse that's down in the lower cord, that's going to be processing information in a much more complex way. It's not just going to be generating, it's going to have IPSPs and EPSPs generated on it. So I'm not able to link this up to what you're saying, except that there is directionality of flow in tracks. But yet, in spite of the fact that one given track might be pulling all the information down and out to the muscle, for example, it's still going to involve EPSPs and IPSPs wherever there's a synapse.
So I'm thinking that that's necessary for the cell to go back to its normal resting state.
No, I don't think it's that. I think that
Oh.
whenever you have a synapse, we conceptualize it as a simple thing. This motor neuron that I'm describing is just passing on information to the next neuron to go out to the muscle. And then that's an EPSP. It would have to be a really big ESP to get the axon hillock to be, but synapses don't work that simply. That motor cell, that lower motor neuron that's going to go out to the muscle has a cell body, and it's got dendrites, and it has thousands of synapses on it. Some of them generate EPSPs and some of them generate IPSPs. In other words, anytime you see the synapse, it's going to be that way, because even at that very kind of basic level, you think this cell communicates with that cell, information is getting processed.
Good. Thank you.
Okay. We both just gave up, didn't we? I think that's.
Yeah, no, it's a great thing. I just didn't know how our body was working, all these systems. And it's so amazing that it does all of these complex things so quickly.
It is amazing. Oh, it is totally
And
amazing.
we're
I mean, you think,
deconstructing
well,
it to a minutia.
We're just trying to get our motor cortex to tell our muscle to contract. What kind of information processing do you need? Well, something like the little muscle fiber next to it, you better contract too, but the one over there don't contract. So, we've got to have EPSPs and IPSPs in order to tune up the movement.
Professor, that's kind of what I want to lead up to. So what is important about the IPSP and the EPSP, I think, the excitatory and the inhibitory? What's like the biggest takeaway that we need to really capture from that last slide?
Well, the biggest takeaway is that neither one is by itself capable of generating an action potential, but they do integrate the information that's being generated in the form of a graded potential on a second order neuron or the next neuron in the chain. They integrate it, and if these conditions are such that it will generate an action potential. So hopefully that's going to make a little more sense. Let's keep going and then come back and look at it again, because I think just every step we take will generate maybe some questions about the old stuff too that we've already talked about. And that's just fine, because you're integrating information. Okay, now yes, I did the EPSPs and the IPSPs. Okay, now this is kind of a simple slide, and kind of simple looking, and it's a slide that could be. In fact, I think I might have had another slide in there and took it out earlier. I know this is the copy that you've got, but we could go into a lot of detail here, but it's not my intention to do it. So here's what I want you to know about an action potential. It is a self-propagating impulse that travels down the length of an axon that has reached threshold. And what's threshold? Minus 55 millivolts at the axon hillock, and that depolarizes each successive axon segment to minus 55 through current flow. Now that's a key part right here. We didn't have this little phrase here in reference to graded potentials. What I said about graded potentials is that they dissipated the further away from the origin you go. They dissipate, and they don't self-propagate. They self-propagate, but only for a very short distance, and then they dissipate, peter out, and they're gone. And the membrane restores itself. It gets all the ions back on the right side and everything's back to resting potential. Well, with this, something kind of amazing happens if you can get a cell to minus 55 millivolts. What happens is that the floodgates to sodium totally open up. It's no longer graded. It just opens up and lets them flood in. And when they flood in, they get the next segment adjacent to it in every direction, down to minus 55. And that segment does the same thing, totally opens up the floodgates to sodium, and that gets the next part and the next part and the next part. So it is a wave of a dramatic increase in the permeability to sodium that travels down and keeps itself going, because it keeps keeping the next part minus 55 and the next part and the next part. Does that make sense what I'm trying to say? It depolarizes each successive axonal segment to minus 55 through current flow. Now there is current flowing, and the current flowing is enough to get it to minus 55. You okay? Okay, let me go on. We can always come back. Okay, now, I made this point in the previous slide, I think, but here it is again. Temporal and spatial summation of graded potentials, that is the EPSPs and IPSPs, occur at the axon hillock. And if the sum reaches minus 55 millivolts, an AP is generated. I'm not sure if I used the term, I think I alluded to it, but did I use the terms temporal and spatial summation before this? Okay, so spatial summation has to do with how closely you get these things together on the same neuron. If they're too far apart, remember, graded potentials dissipate with distance. If they're too far apart, they do not add each other up. They don't add. But if you put two right together, that adds up to twice as much as one. What does? The change in potential across the membrane. So that's our spatial summation. Same thing for temporal. If you get a bunch of graded potentials being generated on a post-synaptic cell in a short amount of time, you're much more likely to get an action potential than if they're spread out. You could have 100 graded potentials, but if each one happens one day, it has totally no effect, except you get a graded potential, but never an action potential. If on the other hand, you get 100 in a half a second, you probably get the axon hillock to threshold. Now that assumes that the graded potentials we're talking about are heavily weighted in the direction of EPSPs, because IPSPs cause an action potential to be less likely to occur. If you got an IPSP and an EPSP generated simultaneously right by the hillock, they would cancel each other and you wouldn't have an action potential. If, on the other hand, you have a whole bunch of EPSPs generated right by the hillock and close in time and not many IPSPs, then you get your action potential, as long as the numbers add up. But that's the principle. Okay, so we got
temporal and spatial have to be, they have to align in order for it to become an action potential.
Yes, one way or the other. You have to get enough excitation going on. You have to get enough EPSPs generated in a short enough amount of time to get that axon hillock to threshold. So if they're too spread out in time, or if you have more IPSPs than EPSPs, it's not going to happen. If you have the required amount, but they're too spread out, the required simultaneity, but they're too spread out, not going to happen. If they're very close in time and enough EPSPs, not too many IPSPs, close to the axon hillock, then you'll get your action potential. All right. Again, we just see the potential for tremendous complexity of information processing when we're talking about that much complexity. And we're only talking about 2 cells right now. Now take that to 80 billion, and it's amazing. All right. Now, what's this next point? Reaching threshold causes the neuronal membrane to have a temporary but significant increase in permeability to Na+. I did say that. And it diffuses inward in response to both the concentration gradient and the electrical gradient. We'll look at the next slide. And until the cross-membrane potential is reversed. Okay, now we'll have a slide that looks a little bit more at the changes, actual changes in potential, and I think that might be the next one. Then what happens? What happens is you get the action potential. It travels down the axon and it propagates itself. And it gets down to its bouton and it secretes this the transmitter substance, whatever that is for that cell, which in turn generates an iPSP or an EPSP on the next cell. Okay, so that's kind of the pattern. But coming back here to our points, the sodium potassium pump goes to work quickly to restore RMP. So once you've generated that action potential, that cell pumps everything back where it's supposed to be, and now the whole cell's at minus 70 millivolts. Meanwhile, an action potential has been generated and travels to the neuron's bouton or boutons, reminding us that any one neuron actually probably has more than one bouton. It's got like a little tree down there at the end with a whole bunch of branches, and each one has a bouton. And
Professor,
yes,
is the sodium, is that the purpose of the sodium potassium pump, to restore the resting membrane potential?
and to keep it there.
To keep it, okay.
Yeah. Yep, that's exactly what it is.
Professor, I have another question.
Okay, sure. I'll go back to that slide first.
You did one of the differences between graded potential and action potential is that graded can like dissipate, but action potential, like the whole all or nothing, it keeps firing. But does the action potential like eventually dissipate in some way, or is it just
Nope.
like
It
converted
goes
or
down to the
recycled?
end. It goes down to the end, and when it gets down to the boutons, one or more boutons, then it causes those boutons to do what we looked at that last week, where the little bouton migrates to the presynaptic membrane, fuses with it, releases its chemical into the cleft. The transmitter then diffuses across the cleft and has its effect of generating either an iPSP or an EPSP on the postsynaptic membrane. So no, that AP is going to go all the way. It's not going to stop.
So it's pretty much a continuous, like it just keeps going, like a cycle.
Yes, and that comes back up here to where it says it's self-propagating. Where is that? Well, it doesn't. Yeah, it's self-propagating. And how is it? Why is it self-propagating? Because it has sufficient. It reaches threshold, and then that brings the next segment to the minus 55. And what's so key about the minus 55? When the potential gets to minus 55, it dramatically increases the permeability to sodium, which then dramatically enters the cell and drives it away from minus 70 and towards 0.
Making that potential, is that right?
So it's using the potential.
Using, okay.
And then the sodium potassium pump is going to restore the potential, because you got to, in order to have any potential to do all of this, you've got to have that voltage difference across the membrane.
Is that the like refraction period?
Yes, yeah. It takes a little while for the sodium potassium pump to do that. And I'm not actually sure how long that is. It's not very long, but there is a short period during which that cell cannot generate another AP, because the sodium potassium pump has not restored the proper differential across the membrane, so it can't do that kind of activity. It's rather short, probably a second or something like that. Okay, now what this is just a representation of the voltage across a membrane across time. So here we have the voltage, here we have time, and here we have at rest. So it doesn't label it, but at rest, we know that that voltage is minus 70 millivolts. And then here comes along an EPSP, and it gets generated. And it goes up. Now, it doesn't go up here. See, that says threshold, and it doesn't show it here, but we know that's minus 55 millivolts. And you see the EPSP doesn't get it there. It doesn't get it there. So it just dissipates, and you return to RMP again, minus 70 millivolts. Okay, now here, excuse me. Here is illustrated a succession in a short amount of time, a succession of EPSPs that are summating. See, there's 3 of them. And there's 3 of them. Here's one, but before that can recover, before the membrane recovers, there's another one. And before it can recover, there's another one. And they're adding up and they get it to minus 55 millivolts threshold. And then here's what happens at that point. The permeability to sodium dramatically increases, in rushes all this sodium, and here is your action potential. And then what happens in addition to the permeability, now we haven't talked about current flow, but there is current flowing in response to this. Even a graded potential produces current, but not enough to get the next segment, the next surrounding area to threshold. Instead, it restores itself and you get back. But there's current flow. And it's actually the current flow that keeps this thing going. If it is a graded potential, it keeps it going, but it dissipates and everything is restored. If it's an action potential, the current flow causes the next adjacent part of the membrane to reach threshold, and then that dramatically lets the sodium in, and there's current flow, and it keeps itself going. And now here, what we see happening is it's restoring itself. The sodium potassium pump is just getting everything back. Meanwhile, that action potential has passed this spot by. This is the measurement at one particular spot. And that's what's taking place across time at that particular spot when an action potential is being generated. And it moves back to rest. And then there is even an overshoot, and then finally a restoration. This is the refractory period here. Yeah, the relative refractory period. I'll just mention how it works if an IPSP were generated. If you were measuring there and an IPSP were generated, the little green line would go in the opposite direction. It would go further away from threshold. So it has exactly the opposite effect. If you were adding up IPSPs and EPSPs and you got enough EPSPs in a short amount of time and not too many IPSPs, and they're kind of spread out, you could get that segment to threshold, and then this would happen. Okay, so this doesn't show IPSPs, but the principle is the same. It just goes in the other direction. But if you get an action potential, it's always going to go up on this particular graph. It's always going to depolarize. It's going to become less polar than it is at rest, which is minus 55, or minus 70, excuse me. Okay.
Question.
Yes.
Would you need multiple IPSPs as well?
Could you? Oh, yes, definitely you can have multiple IPSPs, and they summate too.
Okay.
Yep. Mm-hmm. Okay, now this was just allowing for a review of what we just went through, but I think we probably are okay with this now. We'll come back to it if you need to. Oh, we're almost there. Whoa. Thank goodness. Okay, now, what we're going to talk about now is saltatory conduction. And saltatory conduction is what happens in myelinated fibers that allow the action potential to move down faster. And this shows the picture of myelinated, and it defines saltatory conduction. These things are called nodes of Ranvier, the little sections between the myelin. They're nodes of Ranvier, and they're very narrow, but they're little spaces. And what happens in saltatory conduction is you get the action potential being generated right here at the axon hillock. And instead of propagating itself all the way down at the speed, at its physiological speed, which is, it's got all those things to do, all the permeability changes and everything. It has to do all those things, but it only does them at the nodes of Ranvier. What happens in between is current is flowing and it jumps. And the current comes over here and it depolarizes that little area of membrane to threshold. And it does all the things we went through. And then the current flow goes to the next node of RON-VA. So if you measure how fast this AP is moving, it's moving at its own speed of which is physiologically limited. How fast can that membrane do all these things? It has to do it all the way down. If it's myelinated, it has to go through all those physiological changes in permeability, et cetera, right there at the node. But then once it gets to threshold, you get the current flow, and the current jumps to the next segment and brings it. So it goes faster. How fast does electricity go? It's about the speed of light. So your action potential will go at its physiological rate within the nodes, but at almost the speed of light between the nodes. Why? Because it's electricity is carrying it down. If you get a voltage difference, you get the flow of electricity, and that's really what's happening. It's electrical flow, and it goes as fast as electricity. So saltatory propagation is much faster than non-saltatory. I kind of mentioned this before, that our pain fibers are non-myelinated, and that is why you perceive pain a little bit after the actual event that would cause the pain, because it's being carried by these neurons that have to they have to carry that action potential all the way down without the added benefit of the myelin allowing electrical current to jump from node to node and depolarize just those segments instead of the whole thing. They are found in motor cells of the PNS for striated muscles, fine touch and proprioceptive cells of the PNS, and many neurons also in the CNS. But note, I didn't say pain and temperature cells because they're not there. And so anyway, that's what saltatory conduction is. What does it do for us? It allows for faster propagation of action potentials. And that might actually be, oh, it's not quite the last slide, but it's the last part of really that hardcore physiology, neurophysiology of APs, action potentials, and synaptic transmission, and so forth. So we kind of change a little bit direction here. So if you have any questions, this would be a good time to go over any of this, but we could go over it later too as well, because we have 2 hours.
I was gonna ask, how much electricity do we carry? Like a battery's worth, or?
Oh, that's a good question. Well, yeah.
What can we power? Can Uncle Fester
Like at
really
least
come
turn on a
alive and do the
flashlight or
light
something
bulb?
like that? Can we turn on a flashlight?
Yeah.
How much electricity can our brain produce? Well, somebody somewhere, I guess, could answer that question. I cannot. My sense is not a whole lot, because I've never heard of anybody getting a shock from somebody's brain. So I don't think I think it's enough when you're talking about millivolts, that it's pretty small. It makes those neurons work, but I don't think it's gonna, you're not gonna be able to charge your car with it or anything. How's that for an
I
inadequate?
was just curious about how much electricity do we conduct?
What else could we do?
that.
Practical.
That's-
What else is this thing practical for? Other
Yeah,
than
this is cool.
can we possibly get any other use out of our brain other than just thinking? Okay. All right. Well, listen. We only have like 3 or 4 more slides, and the subject is somewhat different. So why don't we go ahead and take a break, and then we'll come back to those 3 or 4. We'll finish that, and then we'll be ready for whatever we need to do in terms of questions about anything. And I'll have some questions for you if you don't. So, all right, see you back in 10. Before I forget. Here's our shared screen. And there we go. Okay, now these are the implications. And you'll be very happy to leave RMPs, EPSPs, IPSPs, and APs behind for a little while, I guess. All right, what are the implications of all of this? Okay, so we've gone through all of these connections among neurons, and what are the implications basically of all these connections? Here it is. All of our neurologic functions are either hardwired connections among neurons or connections that are acquired on the basis of experience. So which ones do we have hardwired? Basically, whatever you can do at birth is pretty much hardwired. And that would especially, of course, include vegetative functions like your digestive system, something like breathing. Now, breathing you don't have any experience with until the moment of birth, but it's wired in there. The whole system is wired in. So all the synapses have been formed that are necessary for that system to function. Whereas higher cortical functions like language, memory, perception are genetically programmed to some extent, some of them, but they are largely dependent on experience. And what that means is that as we have experiences, we are actually forming new synapses. We grow new synapses. If you look at a brain of a baby, it doesn't have quite as many neurons, but it doesn't have as many synapses at all. And so as you're learning things and just experiencing things, you're actually forming new synapses. And the nature of those synapses dictate how you're going to speak your language, what your words are going to be, the meaning of the words, how you code, all that. It's all based upon experience. So that's actually our learning is the result of forming new synapses. It's kind of amazing, isn't it? Yeah. So your brain is different from my brain. All of our brains are different. Not the gross anatomy. I mean, we all have a premotor strip and et cetera, but the actual wiring is different, most different out in the cortex, because that's where we do our learning. Okay. Genetic program versus experiential based development. Our neurologic functions, including our language, memory, perceptions, feelings, and emotions, are all functions of a pattern of connections among these 50 to 100 billion neurons, each of which has thousands of connections. So that's just a reminder of what you already knew. And there is this this really nice video. It's not very long. It's maybe 15 minutes long or something like that, by a psychologist at UCLA who has written about the influence of experience on these connections, and particularly his area of expertise is about infant experiences and how those first experiences set up your brain in such an important way. There's all kinds of studies that try to measure the number of synapses based upon experiences, where the synapses are growing based upon experiences, et cetera. And so it's just a really, really fascinating video, easy to follow. And I would suggest you like it. It's relevant to us in, child language. Well, I say us, I'm not in child language, but it's very relevant to understanding acquisition of language and acquisition of social behaviors, acquisition of ability to bond, etc., based upon those early infant experiences in the first 2 or 3 years. All right. So there was just 2 slides. That was it. And then we can go into several questions. And please ask questions anytime you have them. So you can just interrupt me. If I don't see your electronic hand go up, which I probably won't, just unmute yourself and ask. Okay, so I think you're going to know this one. The point of contact from one axon to another neuron is the what?
D.
E.
It is the bouton, the terminal bouton. That's the point of contact. Yep. Yeah, it's at the synapse. And the only part of the synapse is that is mentioned here is the terminal bouton. You could also say a point of contact would be the postsynaptic membrane. You need a presynaptic membrane, you need a postsynaptic membrane, you need a bouton. And so of the things that are here, that's a point of contact from one axon to another. Make sense now? These things are, well, that's just the plasm inside of a neuron. Every cell has a plasm in it, and the little organelles are kind of floating around in it. It's just like cytoplasm. Endoplasmic reticulum is a is one of the organelles inside of most cells have that. And then a neurofilament is another element of that provides some structure and provides for movement of things around the cell inside, but it's the terminal. During
Professor,
yes,
on question 1, where we said axoplasm, on figure 4.2 of our book on page 76, they have the axoplasmic transport. And it says substances required by the axon are delivered from the soma via antegrade transport. Retrograde transport moves substances from the axon to the soma. And the
okay.
proteins walk the vesicles along the microtubules, and that's shown in red.
That is,
So
are you the
anyway,
one
I
who,
was
are you the one
thinking
who was asking that question
about those highways.
those anterograde and retrograde?
Yeah, that's what
yeah.
I was thinking about.
Okay.
But I was thinking,
Yeah.
oh, that must be just for the cell now.
Oh, okay. Now, okay, so now let me just give a stab at my own rendition of what those things are. You do have, the cell has to move materials around inside. For example, just to manufacture, the actual manufacture of a transmitter takes place up in the cell body. And then something has to get it down there. And what would do it? Anterograde transport moves those molecules down there, and then they get assembled inside of a vesicle.
I see. Thank you.
So
for
if every cell has that things have to be moved around. Just think about it. Like that.
Got it.
Yep.
So
okay.
that had nothing to do with efferent and transmission.
No, it did not. No, that simply
Oh.
had to do with maintaining the vitality of the cell
God.
and its certain functions. Because I brought up transmitter, and you could say, well, that's not essential for the vitality of the cell. It is essential for the cell to do what it's supposed to do, and that is generate this graded potential on the next cell. And you got to get some chemicals down there to do it.
God. Thank you, sir.
Oh, you're welcome. I'm glad we figured that one out. Okay, during resting potential, well, I could say during resting membrane potential, probably better. Which one of these? The extracellular fluid is 70 millivolts more negative than the cell cytoplasm. No, that's not true, because it's the intracellular fluid that's more negative. The cell is firing an impulse. No, that's not true, because it's resting. It's not generating an AP. The cell is actively responding to impulses from other cells. Well, yes, I would say that would be true. So let's hold C there for a moment. And the ionic difference across the membrane is at a steady state. And that is true. Oh, okay. So yeah, see, I'm not thinking. If there are graded potentials being generated, that would be true, but that's not what we're talking about. We're talking about the resting membrane potential. So it is D. The ions, the sodiums and the potassiums, and there's a little bit of leakage, but they're being restored with a sodium potassium pump and maintained at that steady state.
So, okay, so that's, I'm confused, because in my mind, I had put none of the above. Because I could not find I was trying to find it and I couldn't figure it out.
Does
Wasn't,
B make sense now, Georgina?
yeah, it does.
Excuse me, Professor.
Yes.
My question too is a little different. It says, regarding the concentration gradient, all of the following are true except.
It's during.
Oh, yes. You have. Where are you getting that from?
Well, I downloaded the
Before.
Well, I didn't know if it was before or not, because
Yes, it was. I just changed this. Actually,
I see.
today. So that's why if you go to Canvas, it will be. Yeah. Oh, that reminds me of something, and we'll come back to this in a moment.
Yeah, because I noticed there were some consistencies with the slides that you had from the lecture.
Yeah. Yes, I took out 3
So I
questions because I felt after getting all done with the, reviewing the lecture, that they weren't good questions. So
Oh,
yeah,
I understand.
that's why.
But there was also some, just for example, on the spinal cord, you had some of the information marked incorrectly on the original slides, and you adjusted it when you did the lecture. So like,
So say that again.
for the coccygeal, I'm not pronouncing, you said you had two.
Oh,
or,
are you talking about
the
chapter
slides.
2?
Some of the numbers were incorrect.
Chapter
But they were
2?
correct on your lecture, but not correct on the slides.
Are you talking about chapter 2?
Yes, I am.
Yes,
I
okay.
figure I would just mention that at the moment.
Thank you for mentioning it. And yes, somebody emailed me this morning. And so I went, oh, what's going on here? And I went, and sure enough, I had revised the lecture, and then I was, the lecture slides. And then I was lecturing with those. So when you look at the recording, I'm using the right ones, but I didn't properly post them back to Canvas, the revision.
Yeah. What I
So-
usually do is I print a hard copy, and I usually make notes on it.
Yeah,
But
yeah.
so I usually print ahead. So that's
Yeah.
probably why it's off.
That's, yeah. And I hope that doesn't happen again, but I can't promise I don't know. Oh, I'll give you an example of what I just did.
And Mark,
just-
the
I
question
just
that
found
he's
another error
using
right
is question 5.
I beg your pardon?
Oh, I was just mentioning to Mark that the question you have posted right now that you're sharing is question 5 on
Yes.
the,
That's right,
on
because I took out 2,
PowerPoint.
3, and 4. Yeah, and then I
Oh,
renumbered.
thank you, Jasmine. I appreciate that.
Yeah. So yes, it is different. So if you're finding inconsistencies in this one, that you have to go to the new one. And I just changed that today. But I did repost this. I did repost it, so they're on there right now, but they weren't right earlier. Okay. Also on the I told you I updated the syllabus, which I did, and I reposted it. However, I don't know if you've ever noticed, but on the syllabus, I have under the title, I have the update date. So we can keep track of what version we're talking about. And I updated it today, but I failed to write the update date on the syllabus itself. So the right syllabus is posted on Canvas, but it doesn't say September 1st. And so I'll go back and change that. And so it will, all things will be copacetic.
Professor, before we move
Yes.
on,
Uh-huh.
okay, for this question 2, can you explain the answer, which is ionic differences across the membrane, is that of a steady state? Is
Oh,
that because
sure.
they're both?
Yeah.
Can you explain that?
I think I can. I'm going to go back to this. And so this is I'm going to go back to this. It's the same picture, but here I'm talking about resting membrane potentials. And the concentrations of the ions are different on the two sides. And in the resting membrane, they're staying constant. In other words, they're not changing. They're actually changing a little bit, but the instant there's a change, it's restored. So you get a little bit of leakage, of potassium out and a little bit of leakage of sodium in, but the potassium pump is constantly operating. Do you ever, if you have ever been on a boat, there is something in the boat called the, what's the pump in the bottom of the boat? Does anyone know? I want to call it a sump.
Oh, the bilge pump?
The bilge pump. Thank you, Mark. That's essentially what the sodium potassium pump is doing. Every boat leaks. Let's just say that. Every single boat leaks through the little holes and inlets, and where the propeller comes out, a little water drips in there. And so over time, and in an older boat, it can be pretty fast. There's water filling up at the bottom, and the bilge pump automatically should come on if it's working. If it doesn't, your boat will sink. And that's what the sodium potassium is doing. So in the steady state, the resting situation on a boat is that the water is on the outside and it's not on the inside. Same here, there's a concentration difference, but there's a little bit of leakage, and the sodium potassium pump restores it. So hopefully that makes sense when we go back here to number 2 then and look at that. The ionic difference across the membrane is at a steady state.
Can I ask, is that when we're at homeostasis?
Well, the homeostasis has to do with more the whole organism being in a whole state of homeostasis, everything balanced. So it's kind of a homeostasis. I guess you could say it's a homeostasis for a neuron. But usually that term homeostasis is used more for a total organism being in some state of balance with itself or with nature.
So when we're in a run or flight or fight mode,
Okay,
we're not in homeostasis.
that would be true.
Okay.
Yeah, that would be true. Yep, definitely. Okay, I'll go on. Number 3, all of the following are characteristic of an action potential except. Well, I'm going to read them quietly to myself. You do the same and let anyone speak up when you think you've got it. Oh yeah. Okay, I know.
You see, I got that one. I was looking, I couldn't find the answer. I was trying to find it and I was trying to make sense of it.
Uh-huh.
Um, you know, I was I don't know if it was B or C. That's where I was confused.
Well, it is not B or C. Does anyone else? Anyone else have of
Have
the
it
remaining
A?
possibility? It is A. Now, Mark, can you explain how you figured out that it's A? What is it about that?
Definitions of the action potential. It's propagated at constant velocity, propagated at constant amplitude, and it begins at the axon hillock, and it functions as an all or nothing matter. So all those are all descriptions of the action potential.
Okay, so you did it by method of elimination, and that left only one, and
Yeah.
and it is also true of number 1. I mean, you can just eliminate B, C, and D and E as you just explained, Mark. But A also is not correct because the action potential is simply goes down to the end and causes secretion of the synapse. There's no size difference in an action potential in any one neuron.
But isn't neural information electrochemical, not by amplitude?
I'll say that again.
But that's sort of how I deducted that A was not the answer, because I thought the neural information was not signaled by an amplitude. I thought it was signaled by a chemical messenger.
Well, because you're saying the neural information is you're thinking about the synapse being signaled, and that is by chemical. That's true. Yeah. Yeah, and so yeah, I mean that's another way of looking at it. But I would also think, well, even if you're thinking about the amplitude, you don't have such a thing as a big action potential arriving down at a bouton and causing a greater amount of transmitter being released sometimes, and less some other times. It's the action potential is what it is. It is constant. It gets down there with its amplitude, and it causes the secretion of the transmitter. The only thing that will cause a variation in how much transmitter is secreted would be if it had recently secreted some and it didn't have as much left. So that'll there's a fatigue effect that would affect how much, but the amplitude of the action potential itself would still be the same. Okay. Neural impulse is transmitted across a synaptic cleft primarily through anyone.
I mean, is that electrical?
It's not electrical. There is such a thing as an electrical synapse, but they're not very common in the system, and it's not what we've studied.
That's the chemical.
It is the chemical. It's the chemical called, well, another name for it is transmitter. And they have different names. Like, have you heard of acetylcholine? Acetylcholine. It is the transmitter that is secreted by the final neuron on the way out to a muscle. And it synapses, it secretes acetylcholine. And so what's that chemical do? It's a chemical. And what does that chemical do? it causes the postsynaptic membrane to increase its permeability to sodium dramatically. Yeah. Okay, that is it. So I'll stop sharing. Well, before I stop sharing, is there anything that you want me to go over on this set of slides again?
Yeah, like for question 3, the one that it was A,
Huh?
where in the power slides, I was looking for the power slides to try to find the answer and I just couldn't find it.
Oh, okay. Neural information is signaled by the amplitude of the action potential. Okay, so we're going to go back here to where we say some things about APs, and it might be here. No, not there. Resting membrane potential. Action potentials, okay. Well, here, if I were to put it, this would be the place to put it. It's a self-propagating impulse that travels down the length. And where someplace it says it's all or nothing. Does anyone know that slide? It's all or nothing. Because I think, Mark, you mentioned all or nothing. Did you get that from a slide or from the textbook? Maybe from the textbook.
I got that from the textbook when I read it.
Okay, from the textbook. Okay, so
Yeah, I couldn't find the amplitude of it. I was like, is it amplitude? Is it velocity?
Yeah.
Is it,
well, it has an amplitude, certainly, but it's always the same. It's always the same. And that's connoted by that term all or nothing. The action potential is all or nothing, and the all is the constancy of the amplitude of an action potential. But if I were going to add it to the slide, I would add it right here. A self-propagating impulse that travels down the length depolarizes and to -55. It is of constant amplitude. Sorry, constant amplitude, and all or nothing in nature. Okay, good question.
and also for the last one, because I was looking for the answer and all I could find was electrical, and I also found like electrical, chemical. I think it was one of the slides.
Mhm.
So I thought because the cleft, I remember you talking about it being salt water. So I thought, is that what he means? Is it chemical? But that's why I was like a little thrown off.
Well, there is both a chemical and an electrical powers going on. So when we talk about the flow of, we'll just talk about the what is pulling sodium into the inside of a cell. What's pulling it in? Two things actually: chemical forces, the gradient, and electrical forces, the charges. Yeah.
So cleft would be the operative word when it comes to chemical.
Say it again.
So cleft would be the one for the chemical portion,
Cleft?
right? Yeah. Cleft.
Well, yes, certainly that's the way the message gets transmitted across that cleft, is by a chemical, not by electricity. Yeah. All right, anything else on the slides before I take them away? Okay, all right.
Are we gonna go through the questions for chapter 2?
Oh, if that would be helpful at this time, if you want to, yes, we could. Because now a lot of people haven't done chapter 2 yet, I would assume. Have you, I mean, it doesn't mean we can't go through the questions, but have you done, and not all of you have done it, right? I mean,
I did.
You did.
Yeah,
a lot of people probably have not, but there's no reason why we can't. They're just there for, illustrating the kinds of things we might ask. But I
don't want to impose on anyone if they don't want to. I understand. I'll talk to you personally. So.
Okay, yes, we could go through them. Sure, certainly. And we, by all means, will go through them next time when we start the class. Yeah. But yeah, we can go through them at any time. It's not a secret or anything like that. Okay. Any other questions about anything? Okay. Well, I was thinking about clinical problems, and I kind of came up with this conclusion. We don't have enough information yet to solve any clinical problems with, at least not much. So I don't think I'm going to have clinical questions for you this time, but next time I will. And so what we'll do is we'll go through any questions you have about chapter 2, and then I will have some clinical problems for you to solve, and not too many. But as time goes on, you know, each week you accumulate a little more information, but you add it to what you already know, and you can solve a whole lot more problems. So as we get to further on in the question, in the semester, problem solving will become a bigger and a bigger part of it. And so just keep up with everything. So you're gathering the tools right now to be able to solve problems. But right now, there are not too many because, well, SLPs don't intervene at the level of the synapse or the chemistry of the neural transmission or anything like that. And that we're not going to solve problems in terms of the theory of localization or holistic function, really. So I think that you know the planes. I could just throw some questions at you. What is the plane that is would be that you'd have exposed if you cut the body exactly into a right and a left half?
Sagittal?
Yes, it is a sagittal, but it's actually just embellished with a little more of a.
Is it mid-sagittal?
Sagittal?
It's mid-sagittal, yeah. And then what is it if it's you separate into right and left, but they're not equal? It's a little off center.
Parasagittal.
Parasagittal, yeah. There was another name for that I never used. I don't remember what it was called. Does anyone remember that? I thought there was something parasagittal, or you could call it something else sagittal. Am I wrong on that? Might be.
Is that the axis or something like that? The axis, thought I saw it. Hold on.
I don't think so. No, I don't think it's that. Anything else? Okay. Well, listen. Let's end early tonight. You can get started. If anyone wants to stay behind and discuss, go over questions from chapter 2, we can do that right now. I do have an office hour, and I don't think I have an appointment. If you want an appointment, you can have it. If you want to just show up, you can show up. And actually, since I don't have anything to do right now, if you hang around, we can cover any kind of question you might have about anything so far right now. So I will say good night, but I'm not going away. And if you want to ask anything, stay around.
Professor, well, I'm gonna ask you some questions because can you go over the three the funiculi?
Oh, the funiculi, of course. Yeah. No, that is from chapter 2, right?
Yeah.
So let me go get my illustrations from chapter 2.
Wait, Professor, I had a quick question that was just not related to that. I just, for clarification, are the quizzes that we're doing from, sorry, Georgina, are the quizzes that we're doing from here on out going to be in class, or this is
Correct.
just an exception for this week?
No, it's always going to be in class.
From now on, it's going to be in class.
Exactly. So you're going to, this week, if you haven't already done it, and you don't need to have, you're going to read chapter 2. You're going to watch the 2 lectures on chapter 2.
Yeah.
You're going to study a little more. You're going to come to class. We will review the quiz, at the end of the lecture, those questions. Yeah. Okay. And we'll answer any questions. I'll probably have a few clinical questions for you, and then we'll take the quiz at 6 o'clock.
Quiz at 6 o'clock.
Yep.
Okay, thank you.
You're welcome. Bye. Okay, let's see. I'm looking for chapter 2, and I'll find the funiculi. The funiculus, the funiculi.
Will you be recording this portion too, Professor, and posting it, or is it
No.
just for only?
Yeah, no, I'm not gonna do, at least I hadn't planned to do discussion. It doesn't seem like a very good use of your time, really. I don't think. Well, let me check and make sure I turned off the
Because I
recording.
did have some questions, clarification